In this lesson, we are talking about pathogenic molecules and virulence factors. So this is in contrast to pathogenic organisms. Here are our dot points. Instead of talking about bacteria, fungi, protists, and parasites, we're talking about prions and viruses. All right, let's talk about the idea of molecular pathogens. So infectious diseases are caused by these pathogens and they can be transmitted between individuals, but pathogens can be cellular or they can be consisting of just molecules, right? So if they're cellular, they can be single or multicellular, but if they're non-cellular, they can consist of simple or really complex rearrangements, of lots of different organic molecules. So we're talking proteins, we're talking nucleic acids. They don't have the cellular machinery needed in order to reproduce independently, so they use ours or any other organisms. Now, an antigen is something that is found on the cell surface of normal host cells, bacterial cells, and can even be part of a molecular sequence. And what they do is they basically trigger the immune system. So you will hear me talking about antigens throughout the time. All right, non-cellular pathogens are generally smaller than cellular pathogens. When we're talking prions, they're super tiny and they are the smallest known pathogens. But if we're looking at virus here, prions fit in as a protein, and everything else like bacteria are generally much bigger than that. All right, let's talk about non-cellular pathogens in the exact same way we talked about organisms, so non-cellular pathogens basically. Right, viruses, number one, they are obligate intracellular pathogens. They are obligated to get inside the cell in order to do their work. And they are called the virions, which is what we call them, contain genetic material and they're enclosed in a protein capsule. Now, this varies substantially. You can have so many different types of viruses, but they're all basically structured in a similar way. They all carry DNA or they all carry RNA, one or the other, and that's their viral genome. And this viral genome encodes instructions for making new virus particles. And the nucleic acid is that bit surrounded by the capsid there. And many viruses also have an extra little envelope as well around the outside. Okay, viruses inject the nucleic acid into the host cell and instructs it to make copies. Essentially, once it's inside, it can't survive, but it also can evade the immune defenses because it's inside a normal cell. So viruses are super specific, and what they can do is release, or they essentially lyse the cell, burst the cell open, so those viral particles they photocopy can go off into other cells and therefore create more and more and more. So they're very highly specific to their host cell and organism. So often they can their genome can change and they can jump between species, but generally it's really important for them to stay within the same species. And you can see there, once the virus has entered the cell, it photocopies itself, essentially reproduces, and then lyses the cell and releases itself to continue that job elsewhere. All right, viruses are spread through so many ways, right? Direct contact, contact with other bodily fluids, so indirect contact. They can be in aerosols, so droplets or airborne. They can be coming through a vector like dengue fever, which comes through a mosquito. So many different ways. There's millions of different viruses, right? Millions. And their structure greatly depends on that virus family, and they vary in terms of the organism that they are affected or capable of infecting. So it's going to vary how greatly they also mutate, right? Knowing that they have a genome there and they can do that. Right, some diseases. Influenza is a viral disease, and you can see each year those numbers can increase. Now, we in Australia had about 400 deaths from influenza last year alone, and it is quite a serious disease. And it is spread via aerosol droplets produced by coughing or sneezing. We understand that, but you can also pick it up indirectly if you're contacting something like a contaminated doorknob and then putting that in your face. We understand that based on COVID at the moment. You can also get direct contact with infected individuals. It's quite a sophisticated virus, really. Dengue fever is transmitted via vector transmission via a vector which is a mosquito, and dengue fever is in the tropics in Australia, but is definitely around that equatorial region there. And it's an illness that's similar to a really serious case of flu, and it can last about a week, but it can also be very serious and fatal. And so we do have quite a large number of deaths at times, in waves sometimes. All right, virulence factors of viruses. Really importantly, they must be able to attach to the host cell because they have to be able to get inside, and they have particular spikes on their capsids on their envelopes in order to attach to specific receptors on specific types of cells. They do then invade the host cell, so they get in via the envelope and they can fuse with the cell membrane and actually kind of get their genome right in there via endocytosis. They also really have to evade the host immune system, right? The immune system is scanning, going, what's here, what's not meant to be here. And basically, because they are hiding within a cell, they are not necessarily out in the open all the time. Also, because they're able to mutate quite a bit, the immune system sometimes can't keep up and it says, oh, I've never seen that before, I don't know what that is, or maybe I have seen that before, and it gets quite confused. All right, so viruses also have the ability to evolve their genome by reassortment or recombination. So the flu does this quite often, and genetic reassortment, so changing around its genes with other versions, means our immune system will not recognize it, and that's why we keep needing a new flu vaccine every single year to really take on board what the changes have been happening since the last flu season. Influenza has these two things, hemagglutinin and neuraminidase, and these are basically factors on the outside of its surface proteins, and they allow the virus particle to be released from the host cell. That's what neuraminidase does, but hemagglutinin is that attachment and fusion factor. So it has a slight, opposing role in getting the virus to make sure it is making us sick. Plenty of different versions of viruses, plenty of examples to choose from there. You can see, again, a really wide range of transmission factors there.